So today we will look at an overview of molecular biology, so we are all on the same page. Some of this will be just review for all of you, hopefully. It's basic biology information. So we'll try to focus on how we can model these different types of biological data on the computer. All right. So let me ask you guys another question. How would you define life or a living organism? It can be as philosophical or as practical as you'd like to be. Yes.
Interaction with the environment.
Okay, so a living organism is something that interacts with its environment. Okay. Reproduce, so that's a very important feature of living organisms. It needs to reproduce. Yeah, good definition. All right. Any others? All of these are good definitions. So living organisms are entropy-fighting machines in a way. All right. So entropy, which is a measure of disorder, always increases based on the second law of thermodynamics. Living organisms, on the other hand, are very orderly, right? They accomplish this at the expense of increasing the entropy on the outside. So how do they do that? The functions of life facilitate acquisition and expenditure, orderly expenditure of energy. And the tasks that they have are gather energy from the environment, getting food from the environment, or solar power, and then use this energy to maintain inside and outside separation. Okay, so the inside-outside separation doesn't come automatically. They have to constantly maintain that separation. And then use this energy to reproduce, produce new copies of themselves that are slightly different at each generation. And then to also develop strategies to perform all of these tasks more efficiently, because the fittest of the survival of the fittest, they end up developing different strategies to compete with others. Okay. To accomplish these tasks, the basic components that are available to life as we know it are these macromolecules. Lipids provide separation of inside-outside, so the cell membrane is made out of lipid. It's sort of an oily substance, right? Oil floats on top. It keeps separate. It keeps water separate. Provides a layer that separates different parts of the environment. And then proteins and RNA perform enzymatic functions or other functions in the cell. Proteins are the main functional. They have the main functional roles, but RNAs can also work as enzymes. Okay. DNA is responsible for encoding the information about how and when these other macromolecules are being produced. So DNA is mostly for encoding the information. Proteins and RNA are for performing these functions that are encoded in the DNA. And the main functionality of the lipids are to provide separation of the cell from its environment. The DNA is transcribed and translated into protein. It's also replicated to produce a new copy of itself. The enzymes involved in these processes are DNA polymerase. So polymerase means you take smaller monomers and make longer chains of something. So DNA polymerase means you produce DNA from individual components, individual nucleotides. DNA polymerase uses a DNA molecule to generate a new copy of DNA. Transcription refers to taking a DNA and producing an RNA molecule. And the enzyme involved for that process is RNA polymerase. It produces RNA as a result using, again, individual nucleotides. DNA is a double helix. RNA is a single chain. RNA is then translated to a protein using ribosome. So we don't just have a single enzyme anymore. Even these are not just single enzymes. They work with a number of other proteins to perform this function. Ribosome is a complex of RNA and protein molecules that sort of provide that machinery that takes an RNA molecule and converts it and codes that RNA molecule into a protein structure, into a protein molecule. Okay, so it interprets the sequence present on the RNA to produce a particular sequence of protein. There are some variations of this central dogma, such as reverse transcription, where a virus especially can use a reverse transcriptase enzyme to take an RNA molecule and generate a DNA molecule so that this DNA can be embedded in the host genome. There are some RNA viruses where the mature virus will have an RNA molecule encoding its genetic information and sort of enclosed in a capsule, in a package. And when that RNA molecule gets into the cell of the host, it needs to embed itself to the host DNA. One step to accomplish that is to convert the RNA to a DNA molecule using the reverse transcriptase enzyme. There are also some, again, especially viruses that do RNA replication, taking an RNA template and creating another copy of our RNA, just like DNA replication. Okay. All right. So those are sort of the add-ons to the classical central dogma. There are 4 or 5 different nucleotide monomers, depending on whether you are looking at DNA or RNA. A nucleotide monomer is composed of a sugar and phosphate backbone and a base. All of the nucleotides, A, C, G, T, as using the short letters, and then in RNA we have the uracil, U. These are the 5 different nucleotide monomers. They all have the same sugar and phosphate backbone. What makes them different is this base right here. Okay. So when the base is, say, this one, it becomes a cytosine. When it's the next one right here, it's a uracil, thymine, adenine, and guanine. Some of these bases have 2 rings. Some of them have just 1 ring. And we have a different name for the ones with a 1 ring. The ones with the 1 ring are pyrimidines. The one with the 2 rings are called purines. Cytosine, uracil, and thymine have 1 ring. They are pyrimidines. Adenine and guanine have 2 rings. They are called purines. Okay. All right. So these single monomers are linked together using a covalent bond, which is a phosphodiester linkage. So one of them connects to the next one, and then connects to the next one, and so on, using their backbone. So the phosphate sugar backbone makes up this chain of DNA molecules or RNA molecules, and then the bases just hang around on the sides. Make sense? All right. There's a directionality to this linkage. If you look at a sugar molecule, it has 5 carbons, and we'll number them starting from 1, 2, 3, 4, 5. So we have 5 carbon in the sugar molecule. When we say 5 prime, it refers to the fifth carbon. When we say 3', it refers to the third carbon of the sugar molecule. Right? The first monomer has a phosphate group on its 5' end. So this is our first monomer. It covalently binds to the next nucleotide using this phosphate group on the 3' end. When you have a long chain of these nucleotides, the first one has a free 5' phosphate group, and the last one will have a free 3' OH group. Make sense? Okay. Move on. The DNA has two chains of these monomers. So the nucleotides connect with this phosphodiester bond, and the bases, the one ring or the two ring base, is just hanging on the side. And depending on what type of base you have these different names: cytosine, cytosol, adenine, thymine, and so on. And they form a double helix based on hydrogen bonding patterns. Okay? So as a rule, a C makes hydrogen bonds with a G, 3 hydrogen bonds, which means it's stronger than A with a T, which only makes 2 hydrogen bonds. Let's look at the hydrogen bonding pattern here. If we just focus on these 4 nucleotides, where you have 5', not exactly the same one, but we go from 5' to 3' on one, and then we go the opposite direction on the other one, 3' to 5' to 3', but they go in opposite directions. Okay. The hydrogen bonding patterns will dictate what letter is complementary to what other letter. Guanine is complementary to cytosine, and then adenine will be complementary to thymine. Make sense? Okay. The DNA polymerase enzyme is responsible for maintaining that match when it produces a new DNA molecule. So when it produces a new DNA molecule from a template, it will make it so that every new nucleotide that's added will be complementary to the template DNA molecule. Right. There are going to be some errors which are useful and important, but we don't want too many of them. And RNA is considered to be a single chain nucleotide molecule, polynucleotide. It doesn't form a double helix, but the nucleotide complementarity is still available. So we don't get 2 copies of the same, complementary copies of the same RNA molecule, but a single RNA molecule will then fold onto itself wherever there is complementarity. Okay. If we zoom in here, this is an RNA molecule, right? Now, one part of the RNA molecule has ACC, another part has UGG. So those triplets are complementary to each other, right? So there's an A with a U is like a T, a G with a C, and then when they are in close proximity, they'll form a hydrogen bond, which will keep them together. All right. Other portions are not complementary, such as here. There's no complementarity, so they don't bind to each other. They don't stay in close proximity. The complementarity of these letters form a 3-dimensional shape for these RNA molecules, which give them their special shape. So here, that on the right, you see a tRNA molecule, a transfer RNA molecule that falls into this 3-dimensional structure as driven by the complementarity of the bases. Okay. So this right here sort of looks like a double helix, like you would see in a DNA molecule, but it's a single chain that falls back onto itself because there are complementary letters that form hydrogen bonds. Make sense? Okay. As I said, the DNA replication takes one DNA molecule and creates another copy of it. So it unwinds, and then a new strand is produced for each of the original strands. And then the DNA molecules, the double helix that you form from the original one, will have a copy of the original and a newly produced chain that is produced by a DNA polymerase. The other copy will also have one from the original and one new strand. Okay. It's a high fidelity operation. There are very few errors. The average error rate is 1 in a billion. In viruses, the error rate is higher. That's how they are able to rapidly mutate by having a higher error rate. Okay? But for us humans and other eukaryotes, we need this mechanism to be very, very accurate. So the error rate is low. We still want some errors in the organism, especially when it's reproducing, so that these mutations can then be either accepted or rejected in the new generation. So you need some variation in order to drive the evolutionary process. These errors are not set in stone. There are mechanisms in the cell that will then go and detect errors and try to fix them. Okay. So even if the DNA polymerase enzyme sometimes makes errors, there are other enzymes that will go and error check what the DNA polymerase has done and try to fix the problem.